Good morning, everybody, and welcome to our last Marine Science in the Morning for 2025. We're very excited to have Dean Janiak from the Smithsonian Marine Station across the street. And he is a biologist with the Marine Geo program, and he's going to tell you more about structural complexity in the ocean.
Thank you. Good morning, everyone. And so my presentation is it's a little bit about the program that I work for, and so some of the stuff that we do at the Smithsonian, and then I'll give you a little bit of ecology, some marine ecology that many of you might not know about, and then we'll go into some of the work that I've done, some experimental work that I've done that should be, I think, pretty interesting. And so first, I wanted to give you a little overview of the program that I work for. So the program is called Marine Geo. It's Global Earth Observatories. It's a relatively new program at the Smithsonian. So we've been working for maybe 12 years now on this program. So we're relatively new. We're still trying to get our feet wet in how we're doing things. And so our phrase, what we like to use is we say empowering team science to sustain ecosystems on the edge. We really like this phrase, ecosystems on the edge. So this means two things. So this means that we work on the coastal zone on the oceans. And so this also means that these ecosystems are on the edge in terms of their vulnerability to human impacts and climate change. And so what we're trying to do, our research is we're trying to study how these ecosystems work. And it comes to a surprise to me that we've been doing work for so long now in the ocean, we still don't understand some of the basic things about how the ocean works. And so this is one of the things that we're trying to do, but we're doing it not just on a local scale, but we're doing it on a global scale. So that's what I'm going to kind of, one of the points that I'm going to try to tell you about during this. We're really interested in how these ecosystems are changing. Right now, there's climate change is pretty impactful to a lot of these organisms that we're working on. And so things are changing really, really rapidly. And so we're trying to learn not only how these systems work, but how to keep them working in terms of climate change. And the things that we can do as biologists to keep them functioning. And then we want to take all of this data that we're gathering and we want to give it to people, conservation biologists or other managers, whether they're state or federal managers. We want to inform them on best practices, how you can sustain some of these ecosystems. And so that's our end goal. So we want to take our data, not only publish it and make it academically important, but we also want to make it to inform actions of other people so that people can go out to the beach and enjoy the nature there. So this is our domain. These are the habitats that we work in. So we work in emergent vegetation, which is going to be salt marshes, mangroves. We also work in submerged vegetation, so seagrasses, any macroalgae, things that are under the water. We work in hard bottom communities, so that's going to be anything that's like corals or oyster reefs, things that are hard. And then we also work on artificial structures, and that's my expertise. I'm very interested in things that humans put into the water that change the habitat. So thinking about docks or seawalls, when we put them into the water, we get a lot of animals growing on them, and for a variety of reasons, but we're very interested in what those animals are doing and how they're changing the ecosystem. So this is to give you a scope of all the partners that have worked with us over the past 10 years. And so we've gotten to every continent. The red dots are going to be the dots those are the partner sites that continue to work with us year after year. The yellow dots are partners that have worked with us at least one time on different projects. And so we have a pretty big scope over the past 10 years. And so we've done a lot of really interesting work, but I think the take-home point here is that we're a global program. So we're not just doing things in Florida or in the lagoon. We want to think larger. And so we're trying to ask these simple questions on a really large scale. So working for the Smithsonian, we really have three superpowers that we're really, really good at. The first one's going to be long-term monitoring. I know this isn't the most exciting part about doing science, but it's really informative. It tells you who's changing, tells you what's changing, And so it's just tracking change over time. And so these are going to be some of the habitats that we work in. We work in seagrasses, coral reefs, oysters, things that I've kind of already mentioned. And we also have protocols on how you study these types of habitats. And so I've spent a lot of time writing these protocols, how you study seagrasses in the lagoon, but how you also do that in San Francisco Bay or in Chesapeake Bay or in Hong Kong or in Australia. And so that's difficult to do because there's different species, they live in different places, but we want everybody to be doing the same thing. So we really like standardizing these protocols so that when we want to think about seagrasses or corals or oysters on a really large scale, all the data that we're collecting tells us the same story. We can look at it the same way because it's collected in the same way. Our second superpower, and this is probably the most exciting thing, which is I'm going to spend a little bit of time here talking about, is we really like to do these coordinated global experiments. This is something a lot of scientists don't get the opportunity to do. A scientist at a university will do an experiment in their backyard or wherever they're local to. What we like to do is we like to do that same experiment, but we like to do it all around the world. So we're asking a relatively simple question, but we're doing it everywhere so that we can look at how the ocean is functioning in general and not just locally. And that's something that a lot of people don't get the opportunity to do, but it's something that we're really, really good at. We really like to take advantage of it. Our last superpower is with all of this data, we need to figure out how to store this data, how we can look at this data, how we can share it with other people. So this is just data storage. It's technical, it's challenging, but we've gotten pretty good at it. So we have places where we can go now where we can just pull all of this data off and look at it and plot it and graph it and ask really simple questions. And so our partner sites have the ability to take data from all over the world and start to look at that data. And then we're also good at synthesis. And so that means taking that data, telling a story about it. So what's going on? How are these systems changing? What are the consequences of this change? And so that in turn gives us our actionable science. And that tells us what we need to do to prevent some of these things from happening. So I tend to have this in all of my public talks, and I think it's really important because our focus is on biodiversity. And so that's what we're really, really interested in. But a lot of people don't really have a good idea of what biodiversity actually is. And so it's relatively simple. We hear about it all the time. But as a scientist, we tend to call biodiversity different things. Some people will call it just DNA, so genetic biodiversity. So you can look at one particular species and you can look at its genetic diversity, and that could be biodiversity. A lot of people study that. Another form of biodiversity is functional traits. And so herbivores, carnivores, filter feeders, those types of things. That's another form of biodiversity. I think what we're most familiar with, and what I'm going to talk about today, is just species biodiversity. So the number of species you have in a particular place. And so all of this relates to how we have our healthy ecosystem. And so I really like to use a house as an example because we all have houses. And the way that I like to think about biodiversity is think about all of the different nails in your house. They're all different sizes. They all hold different parts of your house together. If you start to lose those nails in your house starts to become vulnerable to storms, or over time it starts to break down. And so this kind of gives you an idea of why biodiversity is important. We want to have biodiversity because it keeps our house or our ecosystem healthy. So what is a healthy ecosystem? It's relatively simple. It's a place where there's very little human impact. And that's getting harder and harder to find these days. People are finding plastics at the bottom of the Mariana Trench, for example, the deepest part of the ocean. There's plastics down there. So it's really hard to find a place where there's very little human impact. Even on our beaches, you can go to any one of our beaches, walk for 5 minutes, and you're going to find trash on those beaches. So it's becoming more difficult to find. That's problematic for the kind of work that we want to do. Another facet of a healthy ecosystem is it being biodiverse. And so you want to have a lot of species in there, but that's also relative to where you are in the world. There's a lot of places, like the desert, for example, that just naturally doesn't have a lot of biodiversity. But in those types of places, you want the animals or the plants that are there, you want to have a healthy population size. So you don't want to have a lot of rare species. There are a lot of rare species, but that's for a variety of reasons. So when you have your healthy ecosystem and you have a lot of biodiversity, why is biodiversity important? Well, it provides a lot of resiliency. And this is basically a fancy word for a place being, it can withstand a disturbance. And so there's, and I'll show you a few slides while this is important, but for instance, well, I'll go through an example in a second. But biodiversity also supports ecosystem functions. This is just the things that an ecosystem does. So for instance, nutrients recycling. So this is basically decomposition. So it's taking big compounds and breaking them down to make them usable for microbes or plants. Primary production, so plants growing. And so another thing is consumers, so predators and herbivores. And these are all really, really important to have in your ecosystem and to make it functionally well. I'll show you a couple examples of those. And lastly, ecosystem services. And this is a word that we like to use, and this is basically what an ecosystem provides to humans. It's the benefits that we get from an ecosystem functioning. And so that could be food, clean water, oxygen, for example, wood, things that we typically take advantage of, we don't think about. These are what the ecosystem provides to us to make our lives a little bit easier. Lastly, carbon storage, and that's going to be a pretty big one, especially in terms of climate change. And so plants, for example, pull CO2, carbon dioxide out of the air, turn it into oxygen. If there's a lot of CO2 in the air, like what's going on now from pollution, industry, the light coming in from the sun can't escape to the earth is basically getting warmer. And so that's a very simplistic view of it, but a healthy ecosystem can help with carbon storage, getting that carbon dioxide out of the atmosphere and burying it somewhere. So what affects biodiversity? So we like to call these drivers, or we like to call them stressors. And so these are sort of global stressors. And so what makes our healthy house and turns it into a degraded house? And so thinking about what turns our ecosystem into a degraded ecosystem. And temperature is a big one, especially in the ocean. A lot of these animals are really, really sensitive to changes in temperature. For instance, coral reefs, they're a few degrees and they're going to start to bleach. And so it's really, really important that we think about temperature as a stressor in these habitats. Habitat loss, I think that's a pretty obvious one. If you're losing your habitat, it's a stressor on the system. Eutrophication, which is basically just extra nutrients in the system. And so fertilizer going into the lagoon is a pretty big one. Non-native species is another one. You're changing communities in there. Non-native species are generally pretty competitive, so they can outcompete native species and change the species there. Increased storms is a big one. So a couple of hurricanes every once in a while is okay, but if you get hurricanes year after year after year, your ecosystem is going to get degraded and it's not going to be able to bounce back. Lastly, overfishing is just an example of where you're losing your species in your ecosystem. And so that's going to have a lot of cascading impacts on what's there. So how do we measure biodiversity? As a scientist, when we go out into the field, how do we do this? It's actually incredibly simple. So we really just pick an area of size that we're interested in. And then we just count the number of species in there. So one of the things that we do annually for the program is we monitor oyster reefs in the lagoon. One of the things that I've developed are these little bio boxes. And this is this white bio box that we put in the oyster reef. When we put it out there, maybe a month or two before we actually go out there and start counting things, and we collect all of the different animals that are living in there. And we identify them. And so we get an idea of how that oyster reef is providing habitat, providing refuge to all of these other animals that are going to be important for a variety of other reasons. So that is one way for us to measure biodiversity. It's just counting the number of species present, but you have to know your area of interest. Another thing that we like to do is we like to use these, what we call them ARMs, so they're autonomous reef monitoring structures. And it's basically just a bunch of PVC plates that we put out in different places around the world. And so we put them out for a certain amount of time, we collect them and we count the number of species in there. And it gives us a really, really good idea for the biodiversity. It doesn't get all of the species, of course, but it's a standardized way that we can use to count what's there. And we can compare different regions or different depths, or we can compare anything, just using something standardized like this. So there's one of the things that we like to do as an ecologist is we like generalizations. And so when we think about global patterns in biodiversity. Is there a pattern that we can find around the world? And there is, and it's actually quite interesting. And it's one of the earliest patterns that ecologists have found, even dating back to the late 1800s. And so there was a gentleman, Alexander von Humboldt, who first noticed that there was a pattern in where species are around the world. And so this top graph is a graph of the total number of species in the ocean. And it's not completely accurate, it's from different studies. But if you go from the poles to the equator, you can see that that line goes up where the equator is and goes back down. So it shows you that we have a latitudinal gradient in where species are distributed. There's more species towards the equators, towards the equator. And so this is going to be pretty important for the study that I'm going to tell you about. Now, this doesn't just happen in the ocean. This happens on land too. And so this bottom graph is from a recent paper where they looked at this. And so this is a meta-analysis, which means it's just taking a lot of different papers or a lot of different data from different authors, putting it together, coming up with a large story. So the first graph is it's a simulation, so it's just a modeling study of all the species, all the terrestrial species, and where we expect to find them. And again, if you're moving from the poles, which is going to be the top and the bottom, to the equator, you can see that we're getting more species toward the equator. And this is actual data for mammals, birds, and amphibians. And so even those groups, we can see a general pattern that there's less diversity at the poles, and as you move down to the equator, you get more species. And so it's one of the patterns that we really like to use to do different types of studies. We can ask a variety of different questions. Now, why this is happening, I can spend an hour trying to tell you why this actually happens. The easiest thing is temperature related is going to be one of the big ones. And so we know that temperatures of poles, as you warm up, you have more species. So the next question is where can you find biodiversity? Is it everywhere? When you go down to the equator, is it all around you? Well, it's not. And so it's going to be distributed in a particular way. And one of the things that we really like to use is complexity. So habitats with complexity, and when I say complexity, I don't mean, what I mean is structural complexity, so physical complexity. So for instance, we like to use structural, topographic, we like to use habitat complexity. This all means the same thing. And so the best example is going to be with a coral reef. And so when you think of a coral reef, you think of all the beautiful fish, all the invertebrates, everything that's around that coral reef. Very simplistic view, but a lot of those animals are there because that coral reef is complex. And so it's got a lot of places to hide. It's got a lot of places where really smaller fish can find refuge. Got a lot of little areas where a lot of these species can go. And so that turns out to be not just with coral reefs, but it's on land and it's on sea too. So for instance, a forest that has a lot of different sized trees will have a lot more biodiversity than the forest that just has all the same size trees. And so having complexity turns out to be really important for biodiversity. And so it's that sort of we're not just looking at species anymore, we're thinking about things in more general terms. Now what creates complexity? Two major things. The first one is going to be abiotic factors, and that just means your physical environment. So rocks, for instance, is an abiotic factor. So having a lot of rocks along the shore, you're going to have a lot of different species in those rocks, so that creates complexity. One of the things that we're really interested in is biotic factors. So that's going to be the plants or the animals that are creating complexity for other animals or plants to live in. So for instance, mangroves, those roots that go into the water, that creates a complex habitat where you're going to find a lot of different species in there. Seagrasses, for instance, create a very complex habitat where you have a lot of juvenile fish that like to hide in there, hiding from the larger fish. Oysters, as I just showed you, is a very complex habitat where you find a lot of crabs and shrimp that are hanging out from the oyster reef, hiding from other predators and making a living there. One thing I forgot to mention is that another abiotic factor that is, within the past couple of decades is going to be artificial habitats. So docks, seawalls, those types of habitats that people are putting into the water that are creating a little bit of complexity that are attracting other species. And so that's what I'm particularly interested in. So I think the next step that we're all thinking about is how do you measure complexity? That's got to be challenging to do, because how do you do it underwater, first of all? How can you get numbers? It actually turns out it's relatively simple. We have simple ways to do it. So the most traditional way that we've been doing it for decades is called a chain and tape method, and it's getting what we call rugosity. And so think about going to a coral reef or a rocky shore or anywhere and just taking a chain of a known length and running it along that area. And so that forms a different pattern in that you can compare the total length of the chain, which how far it goes, and that gives you a simple number to look at, what we call rugosity. And that's been done for forever, and it's well published, it's a well-used way to study complexity in habitat. And it's relatively simple, and it's something that we can do anywhere, and so we like those types of things. Now, there's been a lot of new technology that has come about that a lot of people are really, really excited about, and it's actually quite easy, and this is called photogrammetry. And so the idea is that you can take a drone, or you can take a satellite, and you can take pictures of the habitat below you. You can put it into your computer, and you can get out a lot of different numbers on the complexity of that habitat. And so this is from a study from one of my colleagues where he was looking at the complexity of rocks along the shore. And so they flew a drone over it, then they made a map of the habitat with a lot of different pictures. We can make a mosaic or a composite. And then they put a heat map on there. And this heat map shows you the redder areas are the places that are going to be higher up. So this is what we call a digital elevation model. So it looks at the changes in elevation, and you can take that data and you can turn it into a number that gives you complexity of that habitat. And so it's very easy. All you have to do is fly a drone over an area, and you can get a lot of really, really good information relatively quickly. So this will just give you an idea. So this is some drone work that we've done out of Oyster Reef, maybe a 10-minute boat ride from here. And so you just fly a drone up. This is an oyster reef that we call Jack Island. We can take several pictures from this, and we can put it into a program, and we can make a digital elevation model, and we can look at complexity this way. So we can do it without even having to go out and using this old chain and tape method. And so it's becoming relatively easy to do. And so we can measure the height of the trees, we can measure the oyster movement, we can even measure seagrasses in the water now, just flying a drone for 10 minutes. And so we've come pretty far in the types of things that we can do, and we can automate a lot of these things. So another way that people like to study complexity in the ocean is that we take standardized pieces of PVC and we can manipulate them. So we can make them more complex or flat, essentially. And so this is from a study that was done in several places around the world where they did this. And the bars, each graph is going to be from a different place in the world, and the bars are from flat to complex. Now, a lot of these sites, you can't really tell the difference, but there are some sites that you can. For instance, in Auckland, in the middle, which is in, I think it's in northern New Zealand, you see big differences in the amount of species that you're finding when you change the complexity of where they could live. Some other places like the Chesapeake Bay, for instance, you can see that as you're moving to a flat panel to more complex, you're getting more species, and these are out for the same amount of time. And so this is just more evidence that complexity, wherever you are, is really, really important for the species that you're going to find. So the best marine examples that we have are from coral reefs and fish. And that's for a variety of reasons. Coral reefs are economically pretty important. Recreationally, they're incredibly important. And so these are going to be from two different meta-analyses. So these are just authors taking data from a lot of different papers and putting them together and telling a bigger story. So the first graph on your right is going to be for density and biomass of fish. And on the bottom is your structural complexity. So just think of this in terms of maybe rugosity. The bigger the number is, the more complex the habitat is. And what we're really interested in, and there's going to be several of these kind of graphs, is this trend line. So this trend line tells you what direction you're going when you increase complexity. And so in terms of density of fish, the more complex habitats have more fish. In terms of biomass, again, the more complex the habitat, the more biomass of fish you have. So the more evidence that complexity is really, really important. On this side, they're looking at rugosity using chain and tape method that I just shown you. And they're looking at 3 different depths and in 4 different regions. And the data aren't incredibly clear here, but as you can see, from the 1970s to the 2000s, the complexity of coral reefs are going down. The reefs are becoming flattened. And what that means is that it's going to have cascading effects on all of the other animals that utilize this reef as habitat. And so we know that this is a problem. There's a variety of reasons for that. But over time, we're losing complexity in our coral reefs. So how does complexity relate to ecosystem health? Well, I think I've tried to convince you that the more complex habitat you have, the more diversity you have. And so why is diversity important? Well, as I had mentioned, ecosystem resilience is a really, really big one. And so a lot of these systems, they need to withstand disturbances. So that could be pollution, that could be storms, that could be a variety of things. If a boat happens to go through a seagrass and its prop cuts up a lot of those blades, you want that grass to fill in. So you need to have diversity. I think coral reefs, which I've already sort of mentioned that example is really, really important. So along the keys, continuous storms are going to knock those corals out. If you don't have biodiversity, it's going to be harder and harder for those corals to come back. You need certain species there that maybe are fast growing that can set the stage for other species to come in. And so that's called succession. Food web dynamics is going to be really important. So some examples that we may have heard of in the news, wolves and deer are a really, really good example. So you have a lot of deer, they're eating everything. You need wolves to keep those deer, keep those numbers in check. And so that is a function of biodiversity. An ocean example is going to be urchins and macroalgae, specifically on coral reefs, but this happens non-coral reefs as well, where you can get a lot of algae coming in, but you need those urchins there to eat the algae, keep the algae in check. Sometimes those urchins get out of hand. So on the West Coast, we know that a lot of the urchins now are eating a lot of the larger kelps. The reason for that is because we're losing our sea otters. So the sea otters can't eat the urchins to keep that algae in check. So everything that we're learning is connected. And so there's a balance there. And so for me, the way that I like to think about biodiversity is it brings balance to these different types of habitats. Another important thing about diversity, at least native diversity, is that it prevents a lot of non-native species from coming into your habitat. So if you have, you could have a forest or a coral reef or a variety of different systems where you have a lot of native species. If you have those native species there, that generally will prevent non-native species from coming in. And so there's been several experiments that have shown that pretty well both on land and sea. And lastly, diversity is really important for ecosystem services. So all the things that we benefit from because of diversity. That's sort of a simplistic way to look at it, but there's been a lot of studies that show that. So when we think about complexity, we know that gives us a lot of diversity. But one of the things that we haven't really looked at, which is very surprising to me, is that we don't know if we have more diversity, if that gives us more complexity. So is there a feedback system where you can have a lot of diversity in your habitat, and that's giving you complexity, and in turn it's just sort of a circular sort of idea. And so we haven't done this work yet, and it's very surprising because there's been thousands of papers that can look at complexity and how that supports diversity, but there's been very few papers that look at how diversity can support complexity. And so when we think about this, we can think about this in terms of, say, a forest. We have a monospecific forest where we have just one type of tree, versus a diverse forest where we have lots of different types of trees. How does that support diversity? Well, we don't know. That work hasn't really been done yet. And so this sort of got me thinking, well, maybe we can do this. Maybe we can look at this on a global scale using some of these photogrammetry methods that I've talked about. And so we did what's called a panels experiment. And so, as I had mentioned, I work on artificial structures. And so this will just give you an idea of the communities that I work on. Maybe it's not gonna work. It should be a smooth video. Anyway, so underneath this dock here, which we took this video north of here, but there's all these beautiful different types of animals that many of you probably have never heard before. And these are what I call fouling communities. They're communities of animals that foul different types of hard substrates in the ocean. So there's lots of different species on here. It's very diverse. A lot of them are small, so you can't really see who they are. But this is one of the habitats that I spend a lot of time thinking about. So when I'm thinking about how we can do a project looking at diversity and complexity, this immediately came to mind. And so what we did was we came up with an experiment where we could take plastic panels that are 13 by 13 centimeters, relatively small, put them out at as many sites as we could with people that were interested in doing the work. We ended up having 17 different sites. We hung them off docks, and we looked at the different communities of the animals living on these panels. And so we took a standardized piece of panel, we put it out at all the sites so that the size of the habitat is exactly the same. So we could ask the same question using all of these different sites. And so these are the types of animals that we found in different places. The first one all the way on the right, that's going to be from the lagoon. So these are the species that I work with. This is San Francisco, and this is I think this is going to be in Panama. And so lots of different species, but they're all colonizing these panels, and they do it really, really quickly too. And so the idea was that we wanted to look at how biodiversity affected complexity. And so what we did was we hung these out, and then we took pictures of them at 30 days, at 60 days, and at 90 days. And it turns out that after 90 days, you can get a lot of animals on these. And a lot of them are already starting to die because their lifespans are relatively short. So this is the perfect experiment to do, for instance, during the summer. So it's one of our summer experiments. And so we wanted to do the different ages because things grow, right? So one of the ideas is that, well, if you have an older community, those animals should be growing on there, so maybe they're more complex. So it's another question that we could have asked. And so this will just give you an example of how we approached it. And so, for instance, in Maryland, we have a site there at the Smithsonian in Annapolis, which had very low diversity. And so when we took that panel and we created a 3D model of it using photogrammetry, it's relatively flat. Those animals don't produce a lot of complexity. We compare that to Florida, which there's a lot of animals on there. There's probably at least 30 different animals on this panel, and there's about 2 on there. This is much more complex than that. And so that started to get our wheels turning. That gave us some ideas on, well, maybe this would work. So how did we measure rugosity? And so we did this using some computer software, but we built 3D models of the communities. And so we brought these panels in from the ocean. We just photographed them on a turntable, a lazy Susan. We took a bunch of pictures of them. We put it into a program, and we created a 3D model of its topographic complexity. And then we took rugosity. So we took that traditional chain and tape method, we did it digitally. And so we overlaid 6 different chains on these communities. We averaged those, and we got an idea of what the rugosity was per community for the different ages at the different sites. So what did we find? And so there'll be a few of these graphs. This is for the different ages, so 30 days, 60 days, and 90 days. All the points are going to be individual panels, communities. I'm not going to show you where those panels were from, because that wasn't really our major question. But then latitude is going to go from high latitudes down to low latitudes, so cold to warm sites. And then what we're really interested in is this trend line. And so if the trend line is positive, that means that the more you have, the more richness you have, so that's just the number of species, goes up as you move from the poles to the equator. So this follows the latitudinal gradient biodiversity, which I mentioned as earlier. So we have data that supports that idea. So that's our first evidence that we can maybe look at how complexity has a gradient. And so in all the cases from 30 to 60, 90 days, we found more species were found towards the equator than they were on the coast. Then we wanted to look at rugosity and richness, species richness, so just the number of species. So rugosity, it's on your right, so it goes from 0 to 0.5. Don't worry about those numbers, but that just means that the larger the number is, the more complex these habitats are. And so for 30 days, not so much. We found the line is positive, it's going up with the number of species, but as these communities got older, the strength of that line got a lot stronger. So the older the communities are, we see a really, really interesting trend that the more species that we found in there, the more complex they are. And so what that means to me is that, well, if we have a lot of diversity in these communities, we have a lot of different animals that are different shapes and different sizes. And then when you look at how complex they are, that's creating a lot of complexity. And so that kind of gets to our answer. That's one of the things that we were curious about. And I was really excited to see this result because it's showing that more diversity means more complexity. So one of the things that we like to do is we like to take these graphs and we like to dissect them. So what are all these points telling us? Well, there's going to be a lot of this noise, we call it. So there's going to be these points or these panels that are higher up that don't follow this trend. Whenever you're doing statistical models, you always get, we call outliers or points deviating from your trend. And we want to know why that is. And so we spend a lot of time, we know what the pattern is, but we spend a lot of time thinking about, well, what are those outliers? Why are they there? And so, well, once we started to dig into the data, we realized that, well, these are going to be mid-latitude sites, and they have this particular species, which is called an arborescent bryozoan, and so it grows like a tree. It's an animal. They're very, very common in the ocean, and so, but it's like a tree, and it's very complex. And we find a lot of them in the Mid-Atlantic, but they're pretty dominant. And so, all of those points up there, where we get a lot of rugosity, that's from these particular species that are going to be dominant in these communities. And so they're causing rugosity to increase. So that deviates from our general trend. But that it's really, really interesting because what that means is that if you don't have a lot of biodiversity, you might not have a lot of complexity, but you have other groups of species that are supporting biodiversity in other ways. And that is because they themselves are being complex. So you think about it, for instance, like an oyster reef. There's just one type of oyster on that reef, but it's providing complexity. Well, in mid-latitudes, where there's not a lot of diversity, you have a particular functional group or a particular group that's dominant, that's giving you complexity too. So we found that complexity is it's occurring in two different ways, whether you have a lot of species or whether you have particular groups that are causing complexity. So conclusions from this study, we found that increased diversity gives us increased complexity, and that really hasn't been looked at before. So we're really excited about this research because we think that it can help a lot of different people think about how we look at ecosystems in different ways. And so when we think about diversity, we don't want to just think about the numbers of species. We want to think about their morphology, how they look, how they supporting other species in their different habitats. And so one of the other cool things that we found was that as you move from the north to the south or towards the equator, your complexity increases. And I don't know of any other habitat where you can really look at that. Maybe you can do it with forests, but you can't do it with coral reefs. Coral reefs are only found in the tropics. And so this is one of these very few experimental habitats where we can look at this kind of question, and we can look at it on a global scale. We can make generalizations about complexity along latitude. And so again, in areas where there's not a lot of diversity, we still get complexity because we have certain groups there that have a particular shape that influences. So this is very similar to coral reef studies. So there's been just a handful of coral reef studies that have looked at the different types of species and the shapes of those species, and they found the same exact thing that we found. And so that's really exciting because it's following a general pattern in the ocean. And so we're hoping that with our work, we can sort of inform, the bigger picture on what's going on in the ocean. And so lastly, I kind of want to just put this all into perspective. And so from a conservation point of view, conservation biologists, they're not necessarily trained in going out into the field and doing a lot of monitoring. What they really like are indicators, health indicators. So they like to go out there and they like to get an indication on how healthy their ecosystem is. And so if you're conserving a piece of land, if you're going to buy a land for conservation, how do you study that land over time? Do you count all the species in there? Conservation biologists, that's not what they do. What they're really interested in is just looking at health. So maybe they can use something like a complexity using drones or satellite, and they can use that as a measure, as a surrogate of ecosystem health. Your complex habitat is providing habitat for a lot of other species. And you can do simple experiments where you can count birds, or you can count the number of plants, or you can count the number of insects in these different habitats. And you can show that, well, these complex habitats, these conserved habitats are doing really well for associated biodiversity. So one of the things I've been thinking a lot about lately is complexity a one size fits all? Can that be done sort of in the land and in the sea? And it turns out it really can. After reading enough papers, it looks like that people are finding a lot of similar sorts of things where they can just take satellite imagery, they can look at the plants and the animals in those different habitats, and it seems to be a pretty good indication of the health of that habitat. So going back to the coral reef example, our reefs are becoming more flat over time. Can we measure that with complexity? We can. And so a lot of scientists might be more interested in what species are there, what species we're losing, and how that affects a lot of different things. But from a conservationist point of view, maybe they just want to go out there and use a GoPro and swim over this coral reef and then put it into the computer and come up with a complexity measurement and look at how that's changing over time. That's really, really simple to do. One of the issues that we're having within the past couple of decades is that we don't have a lot of good taxonomists. So there's not a lot of people you can go to tell you what species is this or what species is that. So it's becoming pretty hard to figure out what these species are. And so if you're a conservation biologist, well, you don't want to have to do that. You just want to go out to your habitat, wherever you're trying to conserve, and you want to use a simple measurement to tell you how healthy your habitat is. And so complexity, topographic complexity, can be a pretty good one. And so what our data are showing is that, well, this can be used, and it's relatively simple to use, even for people that are unfamiliar with it. And the nicest thing is that it can be all automated. And so a lot of these, you can take a lot of these photos, put it in computer programs, the computer will do all of it. And so it's becoming relatively easy. And so we're really pushing for these types of indicators to look at how healthy our systems are without actually having to spend a lot of time in those systems, because that's becoming more and more challenging to do. And so sort of the final thought is that complexity, as I've mentioned from all this previous work, supports biodiversity. But what we're finding now is that biodiversity also supports complexity. So there's a balance there, and that's providing a lot of resilience for these communities. And so that's kind of the work that we found with this one project. We want to do more of it. There's a variety of other ideas that we have, and now that we have the tools to do that, we can start to expand on some of this and do some more really interesting work. So that's all I have. These are just some of the animals that we find in these communities. I wanted to just let you look at it. If anyone has any questions, I'd be happy to spend as much time answering those as I can.
I wonder if you can tell us anything about your findings on artificial structures or invasive species and how they play into some of these species.
So, the interesting thing about artificial structures in the ocean is that that's where we find the majority of non-native species. And so, most of the I say non-natives because if I say invasive species, that means that it's having some sort of negative impact on the environment. Non-native just means that, well, it's not supposed to be there. We don't know if it's having a negative impact or not. So we like to use the word non-native until we know that it's having a negative impact. But if you go to a dock piling or under a boat or a marina or a seawall, and you look at all of those animals on there, roughly 30% of them are going to be non-native, and that's higher than anywhere else you could look in the ocean. Most of the non-natives, they're sessile, so they like to live attached to something. So like a barnacle, for example, lives attached to a substrate. Most of the non-natives are going to be of that kind. They come in through boats coming across the ocean. They reproduce in a new area. They start to live on these artificial structures. And one of the things that I spend a lot of time thinking about is where they're coming from and how they're spreading to natural habitat. And it looks like they don't really spread to natural habitat. There are some that do. There's a variety that do, but the majority of them are restricted to these artificial structures. And there's a lot of reasons for that. The biggest reason is that when you put in a new marina, you're putting in a lot of new substrate, hard substrate that isn't really found in the ocean. And so it's providing habitat for a lot of these species to become really, really abundant really, really quickly. Whether or not they move and they have impacts, typically not, some of them do. But artificial structures are definitely a place where we find the majority of non-native species in the ocean.
Now, does that have a negative connotation to the environment or anything like that, or?
Yes and no. And so an artificial structure already isn't supposed to be there. So all the animals living on that structure, it's already degraded in a sense. And so it is having an impact. The artificial structures block light. So if you have grasses or anything underneath there, it's not getting light. A lot of these animals actually, so these are what we call psidians. And so they're really, really common. They're really, really important for fish. Fish love to eat them. They're actually really, really good filters. They filter more than something like an oyster could filter. And they take everything and they keep it inside of them. Oysters are very picky. So when we think about water clarity, a lot of people like to talk about oysters. If you put more oysters in the lagoon, all of a sudden it's going to be clear, which isn't necessarily the case. Oysters are very particular in the size of food that they retain. Something like these acidians, they eat everything. And so they're much better at cleaning water than oysters are. So if you have a lot of these, are they benefiting by water clarity and removing a lot of that extra sedimentation out of the water? Maybe. We don't know because that's really, really hard to measure. But some of them are actually beneficial. Some of them are food. So we do a lot of work with non-natives and fish predation. So will fish eat these? Fish will eat a lot of them. They won't eat some of them, but they're providing food too. So it's a yes and no sort of question or answer rather. You know, it's very species specific and species dependent. And so we're still learning. And there's you know, we work on communities of animals, and then we start to look at individual animals. There's a lot of work that you can do to look at whether they're good or bad, but generally we don't want non-natives here for any problem species.
Now, these non-natives, where are they originating from? Is it through boat traffic?
Yeah, so it can be aquarium trade. So people dump their aquarium out in the ocean because they don't want their aquarium anymore, and then it's spread. So they escape. Yeah, line fish, for example. But on the bottoms of boats, in the ballast water of boats, so the big boats, they fill up their ballast. They're supposed to do it 200 miles offshore, where these species aren't reproducing. A lot of them don't do that. When they travel, if they're traveling internationally, they have to dump them 200 miles offshore. If you're traveling within the country, so domestically, I think it's only 50 miles where they have to dump them offshore. So they're trying to prevent these species from attaching to the coastal zone. A lot of these boats, they don't do that. They're dumping them right in bays because it's easier, and that's where they sit for days or weeks until they get that ship or whatever. So ballast is a really, really big one because the larvae of these animals are in that ballast, or even the animals themselves are living in that ballast. They don't necessarily they don't need light. And so, but even oysters, for instance, on the top, there's one particular species that I've worked on called Didemna, which is it's a colonial acidian, and it's a really bad invasive species, especially in the north. It came in on the top of oysters that were being shipped in from Europe, and somehow it got into the water, and now it carpets the bottom of the ocean. So they can come in a variety of ways, but one of the interesting things about the ocean is that we're never under there. We know we're in the forest all the time, we're in our backyard all the time, we know the plants and animals there, we see changes every day. We don't spend enough time in the ocean. We don't know who's there, we don't know where these things are, we don't know where they're coming from. And so monitoring starts to become really important because you can start to track things. And so with these panels, we put them out every year. We have 6 different sites locally. Every year I see something new. I don't know if it's rare, if it's from here, or I don't know if it's new to the state of Florida. But we're seeing new stuff all the time. And so we track that. Are they getting bigger over time? Are they not getting bigger over time? Where they're coming from, that sort of stuff. So we're monitoring, I call it biosecurity. It's sort of an exaggerated word, but it's really important. And so we put these panels out, and we look at them because they're really tied tightly to their environment, so we can look at long-term changes. But we want to know what's coming in too. And we know that these non-natives really like this habitat, so it's one way that we can see if new ones are coming in. We have several sites around the world now that are doing the same exact thing, so now we can track global movements of some of these species and where they're coming from. Yeah.
Were there any other habitat community types considered for this other than like the farm communities?
So I work with farming communities, and so this is what was my first idea. I was actually watching I really like pyramids, and so when a scientist goes into a pyramid for the first time, they do photogrammetry of the inside of the tomb, and they make mosaics of that. They take all of these types of measurements, and I saw that, and I was like, wow, I can do that. And so we did that with these, and because I work with these, I thought it would be easy. They're easy to manipulate, they're easy to move around, bring them into the lab, put them back out in the field. And so just taking a lot of pictures of them seemed to be a good idea, and they're standardized. So they're all the same, the panel that we use is all the same size, so you can put them out anywhere around the world, and you don't have to worry about any sort of differences in your habitat. And you can also put them out in any habitat. So we put them out in seagrasses, we put them out under mangroves, we put them out under docks. So it just made sense. And also you don't have to get into the water. So you don't need scuba divers, you don't need snorkelers. You can just walk out to your dock, put them in the water, and things grow on it really quickly. And so it just made perfect sense to do this kind of study there.
The ships that are sunken to be artificial reefs, is it possible that they're helping non-native species more than native species?
Absolutely. And so there's been some work in the Keys where they put out these big, they're called reef balls. They put out these big concrete structures that they're using to attract fish. And they're using it for people to go snorkeling around because they produce these beautiful communities. Well, turns out a lot of the animals living on them are non-native, and so they're enhancing that population. When you're dumping a large ship way out in the ocean, there's not a lot of non-native species that far out and that deep, so you're going to get mostly native species down there. But there hasn't been that hasn't been done. So, and I don't know why, but it's just one of those things where we'd love to do it one day, but we haven't done it yet. And so where you can go out and it's relatively easy to quantify the animals on those boats, figure out where they are, who they are, where they're coming from. But that hasn't been done. And so that would change the way people think about whether you should drop a boat out in the middle of the ocean. I personally think they just need someplace to put it. And there's been no strong data that I've seen that suggests you need to put more stuff out there to get better fish populations. And so I just find it a way that they just need to hide the boat somewhere. And they figure, well, we can just dump it out there. I'm not in support of putting artificial structures, whatever it is, out in the ocean. We need to clean it up, not put more stuff out there. And they twist it to make it sound like it's for conservation purposes, but I haven't seen the data. I haven't been convinced yet that it's making an impact, other than providing a place where people can go and scuba dive. And it's yeah, exactly, yeah. Now you know where to go to catch the big fish.
Right.
So that's a problem, it's a problem, because then you lose all your big fish, and then your system sort of collapses. So,
Doesn't that support though the belief that increased structural diversity or artificial?
yeah, no,
I mean,
yeah, you get more biodiversity.
basic diversity.
Yeah, and so everything is a double-edged sword, right? So there's good points and there's bad points, and you have to weigh those. And so it does both. I mean, you're absolutely right. Yeah, a lot of the complexity is really important for juvenile fish, and so you want to have those juvenile fish have a place to hide from the larger fish, so that they'll become large, and so we can keep going fishing. So you'll catch the size of fish that we're allowed to. So I mean, it's all science is difficult, and so there's rules and the regulations, but there's also you have to find a balance of what's good for the ocean, or for the land, and what's not. And we need to use a lot of this data to make those decisions. Yes.
I was just gonna ask how your studies of habitat complexity relate to seagrass and what is going on with our seagrass here.
So complexity is hard to measure in seagrasses because they're the way that they are. We know that the way that they've done it in the past is that they manipulate density of grass. So you can have a dense bed versus a sparse bed versus no bed, and you can compare what's in there. And we know that a very dense bed is very good for fish, for juvenile fish, for all of the smaller invertebrates that live in there. As you start to get thinner and thinner, you get less and less associated diversity. So, so we know that we need good dense grass beds if you want to support biodiversity. It's not a direct measure of complexity, but we can hypothesize that the more dense a bed is, the more complex it is, just by having more sheets in there. So it's sort of another way to think about why you want a dense seagrass bed. It's because it's complex. More places to hide for juvenile fish. Stay away from the larger predators.
So I'm trying to, I'm thinking about the complexity provided by different heights.
Yeah.
Your rotozoans.
Yeah.
Versus complexity that has to do with the variety of species.
Mhm.
I just, it
Yeah.
seems like those are counter issues.
Yes.
Okay, can you
Yeah.
talk about that a little more?
Sure. So when I think of size, I immediately start thinking about forests, because trees are very easy compared to bushes.
Yeah.
So, and then the size of your area is also pretty important. And so a forest, you can pick up pretty large, like an acre, for instance, and you can fly drones over it and you can get a really good measurement. You want a little bit of everything. And I know that's a very simple way to answer the question, but birds are a good example. So there's certain birds that will live high up in trees. There's certain birds that live in low trees. And so if you have one thing that's really high up in the water column or in your forest, it's going to support a certain bird or certain types of organisms. If you have different heights, you're supporting different things. So that's how you increase your biodiversity. We call it niche space. So the more niches you have, the more things will be there. And so some of the earliest work on complexity was actually done by Charles Darwin, looking at finches in the Galapagos. So looking at how particular finches Their beak shape related to the trees that the seeds of the trees that the birds were eating, and so he found that the height of these trees was really important for different species because the different species utilize those different heights. And so if you have one species that's really complex, but it's all the same, it's going to support biodiversity, but it might not support all the different types. You could increase biodiversity by having a lot of different shapes and sizes, I guess is my point. And so you want different heights, you want different widths, you want squares, and you want stars, and you want triangles, and that in turn will give you more species just like those different things. And so that's easy for us to test in the ocean. For example, we could use something like Legos, or just create shapes out of clay. And then we can manipulate those shapes, put them on a panel like this, and put them out and just count the number of animals. And there's been studies that do that. And you can show pretty well that if it's all the same height, you're going to get more biodiversity than if it was just a flat surface. But if you change those heights, then you're going to get a lot of different species because some like it high, some like it low. And so I hope that answers your question, but it's just having a variety of different shapes and sizes and supports more biodiversity.
So it's the concept of guilds.
Yeah,
Okay.
yeah, we could definitely use that word guilds too. Yeah, that fits perfectly. Yeah. Thank you.